The contents is introduction, purpose of study, methodology, results, discussion, conclusion, and reference. The introductions. Genetics is the study of inheritance or the transmission of traits from one generation to the next. There are several modes of inheritance. However, this project will focus only on application of linear algebra in genetics. When two individuals breed, offsprings inherit one allele from each of the parents. We will assume that either of a parent's genes is equally likely to be inherited. The purpose of study. We have chosen to explore how linear algebra can be applied to genetics. More specifically, we will focus entirely on the phenomena of genetics. In order to perform such an analysis, we will use several critical concepts from linear algebra, including the following: difference equations, diagonalization of matrix, inverse of matrix, eigenvalues, and eigenvectors. The methodology. In humans, colorblindness, hereditary baldness, hemophilia are traits controlled by X-linked inheritance, and cystic fibrosis, sickle cell anemia, and Cooley's anemia occurred because of autosomal recessive inheritance. Investigation can be occurred for the propagation of an inherited traits in successive generation by computing powers of a matrix. By calculating eigenvalues, eigenvectors, and matrix rotation, autosomal recessive inheritance diseases effectiveness could be found. For the genetics and for the results, we can see how linear algebra can solve autosomal inheritance. Suppose there is a farmer has a large population of plants consisting of some distributions of all three possible different genotypes. The farmer desires to undertake a breeding program in which each plant in the population is always fertilized with a plant of genotypes, both capital A, and is then replaced by one of its offsprings. We want to derive an expression for the distributions of the three possible genotypes in the population after any number of generations. We can determine the genotype of offspring by genotype of parents. Also, from the table, we can determine the genotype distribution of each generation from the genotype distribution of the preceding generation by the following equations. These are explicit formulas for the fraction of three genotypes in the nth generation of plants in terms of the initial genotype fractions. So, there is genotype respectively for capital A and lowercase a, and for both lowercase a will not be expressed. And for the discussion, the probability of diseases of autosomal inheritance diseases by referring to the calculation of inheritance in which a normal gene capital A dominates an abnormal gene lowercase a. So, according to the calculation of linear algebra, we can see that they have a few probabilities to occur autosomal recessive inheritance. The last one is conclusions. Possible way to control such as disease if for the breeder to always match a female regardless of her genotype with a normal male. In this way, all future offspring will either have a normal father and a normal mother for both capital A and for both capital A matings, or a normal father and a carrier mother for both capital A and lowercase a matings.